Cells of the immune system. In this session, the focus majorly will be on B lymphocytes, a cell type of the immune system which is responsible for humoral immunity. And interestingly, what has been observed is that the B cells are able to uh, be a link between innate immunity and adaptive immunity. So let us look at the learning outcomes of this session. B cells are lymphoid cells that originate and mature in bone marrows in many mammalian systems, and with these playing a major role as mentioned in humoral immunity. B cells on its surface have several characteristic membrane-bound molecules, of which some can act as markers. So when you use the word markers, it is specifically is able to distinguish B cells from all other different lymphoid cells. B cells on interacting with an antigen can get activated to differentiate to give rise to plasma cells which are able to secrete specific antibodies. B cells can, apart from producing antibodies, also have several different functions, of which one of the functions is its ability to act as an antigen presenting cell. And that is also one of the reasons, and it is also part of adaptive immunity. So the adaptive immunity is associated with the fact that it can become very specific in its defense against a particular antigen, and also it can develop what is called as an immunologic memory. Now, the word B lymphocytes has been derived from the fact that it was first discovered in the bursa glands of the birds. Over time, what has been observed is that B lymphocytes are found to evolve from stem cells that are present in bone marrow. And so the letter B still holds true for the lymphocytes. Now, when one looks at development, what is found that the development of B lymphocytes is a long journey from the bone marrow to, which is a primary lymphoid organ, to various secondary lymphoid organs where it is further differentiating to form several subsets. So this is an overall journey of how B lymphocytes develop. So the B lymphocytes begin to develop in the bone marrow, and the bone marrow has stromal cells which can give rise to factors that in fact initiate a series of signals. So the hematopoietic stem cells that are present in the bone marrow are first differentiated into ELP, that is early lymphoid progenitor. The early lymphoid progenitor forms the common lymphoid progenitor, and the common lymphoid progenitor, depending on the factors that are secreted by the stromal cells, can go in for formation of natural killer cells, dendritic cells, and T cells, but they can also give rise to the common lymphoid progenitor 2, and this common lymphoid progenitor 2 gives rise to the B cell lineage. So from the common lymphoid progenitor type 2 cells, one gets what is called as the early pro-B cells, late pro-B cells, pre-B cells, and finally the immature B cells. These immature B cells, as you can see, now have what is called as the B cell receptor present. So it evolves in such a way that it gives rise to formation of a B cell receptor. And B cell receptor is something that is markedly present in any B cell. Now, these immature B cells further move on to secondary lymphoid organs like spleen, Peyer's patches, mucosal associated lymphoid tissues, etc. And in the secondary lymphoid organs, what has been observed is that they form what is called as transitional B cells. In fact, in one interesting fact that has been noted is that of the several different factors that are released by the stromal cells, interleukin 7, which is a cytokine, has a critical role in the B cell development process. Now, when one looks at, okay, before we go into this, okay, From the spleen, one can see that you get several different subsets of the B cells, and the subsets of the B cells further differentiate into terminal B cells that are able to produce antibodies specifically against a particular antigen. Plus, it also gives rise to memory B cells, and therefore you can create what is, or the B cells are able to create what is called as immunologic memory. They're able to remember the antigen for a long period of time, as these memory B cells can last in the system or can last in the body for several years together. Now, let us just focus a little on what happens in the bone marrow, where you have the common lymphoid progenitor 2 cell type evolving to form the immature B cell, and all this takes place in the bone marrow itself. And the passage of these cells from one stage to the other is due to the presence of a microenvironment that contains several different factors that are released by the stromal cells. So, in response to the factors, there are signals that enable the cell to evolve to give rise to different cell types as it progresses. Interesting to note is that with every stage of differentiation, what has been observed that there are specific surface molecules that are formed and seen on these specific cell types. So, for example, the common lymphoid progenitor 2 on its surface has a surface molecule called B220. This also is referred to it is also a form of CD45, and this is something that has been present in all B cells. It also has FLT3 and CD43. Now, as it moves from this to the early progenitor B cells, one can observe that the number of surface molecules has increased, which includes CD10, CD19, 24. This further ongoing further and further, what is observed is that it has a subset of these cluster of differentiated molecules present on the surface, and at the pre-B stage, what is observed is a pre-B cell receptor being formed. This is a beginning of the formation of the complete B cell receptor. And when it reaches the immature B cell, then what is observed is that it has the B220, CD19, 24, CD43 and IgM. So this IgM that you find on the immature B cell is a B cell receptor, and it is membrane bound. So this B cell receptor is always associated on the surface of the, this is not released, this is not secreted, this remains bound to the membrane. Now, this immature B cell further moves on to the secondary lymphoid organs, and when it moves on to the secondary lymphoid organs, there are several chemokines that are present that facilitate the movement of these transitional B cells into different regions. So here this goes into medulla region, and it gives rise to what is called as the marginal zone B cells. So this is one subset of B cell that is formed. Another subset of B cell that can be formed is as the transitional B cells move to follicles, it forms what is called as follicular B cells. So you can already see that both the marginal zone B cells and the follicular B cells, apart from having the membrane bound IgM, also has the membrane bound IgD. So therefore, the B cell receptors that are present on the transitional B cell types can already one can already observe the presence of two types of immunoglobulins on its surface. Now these subsets further move on to get differentiated. So you will see that the marginal zone B cells get differentiated into a larger B cell, which is called as the plasma cell. But marginal zone B cells give rise to a short-lived plasma cell. That means plasma cells are differentiated B cells that are able to secrete antibodies. And these antibodies that are secreted have a very specific isotype, that is, they are able to bind to only one specific antigen. So at this point itself, okay, when you have the B cell receptor being formed, a B cell receptor on a single B cell will always be able to bind to only a specific antigen. But what can definitely happen is that the type of antibodies that are released by the plasma cells need not just be the IgM. So you can see that it is differentiating to give rise to different types of antibodies as well. Like for example, follicular B cells, when they assemble within a germinal center, so a germinal center has several components including macrophages, interdigitating dendritic cells, etc. Within the germinal center, as observed in the spleen and various other secondary lymphoid organs, the germinal center is a microenvironment which is enabling high proliferation of the B cells, and with the high proliferation of the B cells, every B cell within it is able to carry out what is called as somatic hypermutation. And this somatic hypermutation leads to what is called as rearrangement of genes, specifically what is called as VDJ rearrangement, and that gives rise to different antibodies. It gives rise to different antibodies, but with the antibody having a variable region for a specific antigenic determinant. So in effect, what we get within the germinal center is several B cells, and each B cell having a set of antibodies on its surface that is able to bind to a specific antigenic determinant. So at this germinal center, therefore, you would also have what is called as clonal selection. Now, please remember that this will happen only when the antigen is already, the B cell is already exposed to an antigen. So, only on interaction with an antigen, you can have this high proliferation of B cells happening and B cell selection happening. So, the B cell selection happens in such a way that those B cells that have antibodies on its surface that has high affinity for the antigen, only those B cells will be selected. The other B cells will be eliminated through apoptosis. So therefore, such clonal selection takes place within the germinal centers. And what has been observed beyond this is that you have those specific B cells that are selected further getting differentiated into plasma cells. These plasma cells are terminal B cells which can release or secrete antibodies, and they are long lived compared to this one, but they are terminal. They would die within 1 or 2 weeks. However, it also gives rise to, as it differentiates, several memory B cells, and what is observed is that you have class switching happening. So with class switch, what can happen is from IgM you can get IgG, or you can get IgD, or you can get IgA. So different types of antibodies can be formed because of the class switching. So different B cells that are formed within the secondary lymphoid organs, all of these B cells will have a B cell receptor which is very specific or for a certain antigen, and it has a high or a certain affinity. The plasma cells, as mentioned, are terminally differentiated and they die in a stipulated period of time, but the memory cells remain for a longer period of time. Now, what is also interesting to notice that a live B lymphocyte that is not yet associated with antigen, when this associates with antigen, then it induces the cell cycle and it induces the cell division to take place. So what happens is a small live B lymphocyte will increase in size to form what is called as a lymphoblast. In the lymphoblast, you will have DNA synthesis. That cell will divide into two, okay, and of which some cells can get back into the same cycle again and again, while some cells can further get differentiated into plasma cells. These plasma cells are responsible for secreting the antibodies, and some of them will remain as memory B cells. So, a single B cell can have about 1.5 into 10 raised to 5 membrane IGs, that means BCRs, that is B cell receptors. And all the B cell receptors on a single B cell will have the same binding site for a particular antigen. You will not find different antibodies in one B cell with the ability to bind to different antigens. This B cell will be able to bind only and only to a specific antigen. The differentiation into a particular B cell subset, of course, will depend on cytokines, specific transcription factors, etc. So it is a lot dependent on internal signaling as well as external stimulus. The surface markers of these subsets can have variations, and you can demarcate one B cell subset from the other B cell subset depending on what is the range or the different surface proteins that are present. Now, when one looks at the B cell receptor per se, what is very clear is that the B cell receptors that are formed at the pre B cell, okay, has the beginning of the formation of the antibody, okay. So you have the heavy chain with the light chains being formed, and this light chain is still not mature completely. It is still what is called as a surrogate light chain, and it is associated with two immunoglobulin molecules, IgB and IgA. And interestingly, as it develops into the immature B cell, you find that the surrogate light chain becomes a stable light chain, and the heavy chain at its C end, C terminal, is not is bound to the membrane of the B cell through transmembranal segment. So this is about 25 amino acid long segment that remains bound to the membrane, and you have a portion about 5 to 6 amino acids which are intracytoplasmic segment. This part of the antibody or the B cell receptor is a domain where you have variable region present over here, and this variable region has developed by somatic hypermutation to be able to bind to a particular antigenic determinant. These transmembrane segment is able to associate with tyrosine kinase, and for the tyrosine kinase, Ig beta and Ig alpha are the substrate or mediators. So these are signal mediators. When the antigen binds to the B cell receptor, you would have a signal transform, and this in turn will pass on the signal. So based on the signal that is passed on to the B cell will respond accordingly. It will either produce, or it will either give rise to class switching. So from IgM you can have IgA or IgG or IgE being formed, or what can happen is that the signal gives rise to formation of several different cytokines, or it gives rise to formation of several different surface molecules. All those things will happen only when the antigen binds to the antibody. So therefore, the B cell receptor primarily is a membrane-bound IgM or IgD, and it is a monomer. We all know that IgMs that are secreted are pentamers, but when it is present in an immature B cell, it is present as a monomer. So the mature B cell, once you have the B cell maturing, what has been observed is that these are the various different cell surface molecules that are present on a mature B cell. As you can see over here, a mature B cell can have all the different classes of antibodies present, and these are all able to bind to a specific antigenic determinant only. It also has a different class of surface molecules, which are differentiation markers. That is, you can distinguish a particular B cell because of the presence of these marker molecules on its surface. It has class 2 MHC molecule. So the class 2 MHC molecule is able to present antigen on its surface. So, which means that the B cell can act as antigen presenting cells. A B cell, through its BCR, when it binds to an antigen, the BCR antigen complex is internalized by the B cell, and once it is internalized, the antigen is broken down because of the formation of the endolysosome, and the lysosome enzymes are able to break down the antigen into smaller pieces or smaller fragments, and those antigenic fragments are then presented on the surface through class 2 MHC molecule, because you have the B cell having the ability to produce the MHC class 2. So therefore they act as antigen presenting cells. Secondly, because it is a nucleated cell, it can also form what is called as class 1 MHC molecule. And so if the B cell is infected with a virus or an endogenous bacteria, even those can be degraded into smaller fragments, but those are then exhibited on the surface through class 1 MHC molecule. It also has CD40. CD40 is the surface protein that binds to CD40 ligand that is present on T helper cells. So basically, the B cell and the T helper cell can co-stimulate each other by interacting with each other. Then you have several receptors for interleukins present. So if the environment around it has IL-6 or IL-5 or IL-4, they can bind to these receptors. A signal is transduced inside, and based on the signal, the B cells will respond accordingly under or in response to a particular immune situation. Then one has also FCR gamma 2. This FCR gamma 2 is the FC receptor. So it can bind specifically to the FC region of the IgG molecule. So it can carry out a process that is called as opsonization. Because it can bind to the antibody, it can carry out opsonization. It also has receptors for complement system, and complement system is a system that enables to eliminate foreign particles, or to eliminate the antigens. So it is part of the innate immune system, but because it is able to bind to the the B cells are involved in innate mechanism as well. It also has CD80 and CD86, and these two are also able to bind or interact with the T cells. So, co-stimulatory mechanisms are possible because of the presence of these surface molecules. So, apart from just antibody production, B cells therefore have several functions. So, it can act as an antigen presenting cell. So, as it acts as an antigen presenting cell, it can activate a T cell, and these T cells can further get activated and lead to formation of T cell memory as well. Then, because it has surface molecules that can interact with each other, the T cells and the B cells can co-stimulate each other, and thereby resulting in T cell activation and T cell memory, and also stimulation of the B cells per se. Then what is also observed is that the B cells are able to form tertiary lymphoid organs, especially in situations where there is an already existing inflammatory condition. Say, for example, in multiple sclerosis, then you have tertiary lymphoid organs formed thanks to the B cells, and these B cells can create an in situ immune response. Then it is able to release several cytokines, and based on the several cytokines that it releases, it can give rise to a certain specific immune response. Interestingly, what has been also observed is that certain cytokines like IL-10, when released by B cells function as what is called as B regulatory cells. And these B regulatory cells are able to control the inflammatory process. So it can, where at one end it can be pro-inflammatory, at other end what can be observed is, because of its function as a regulatory molecule, it can enable a process or a phenomenon which is called as immunotolerance. And in immunotolerance, it is able to suppress inflammation, or it is able to decrease the antigen presentation, etc. So these things can also be possible by the B cells itself. So therefore, what one can observe is that other than antibody production, these are the various functionalities with which the B cells are associated in various different conditions. So let us make the conclusions. B cells begin to develop in the bone marrow, and with every stage of development, the CD molecules, that is, the cluster of differentiation molecules on the surface, evolve, with the B cell receptor 2 being formed. And this is very characteristic of a B cell. The immature B cells from the bone marrow move to secondary lymphoid organs, wherein they develop into transitional B cells, which can further then form B cell subsets. The follicular B cells move to assemble into germinal centers, where there is increased proliferation of B cells with somatic hypermutation and clonal selection, with B cells having affinity for antigen being selected. The B cell can further differentiate into plasma cells that can secrete antibodies and that can also form memory cells. And this is a hallmark of adaptive immunity. Other than antibody production, the B cells have several other functions that effectuate immune responses. Therefore, B lymphocytes, that is actually bridging innate and adaptive immunity, have characteristic B cell receptors that, on binding to antigen, can trigger a signaling pathway that can lead to prolonged immunity. Thank you.